Hello, I'm Madam Eva, a biology teacher from Educare TZ, and I'm here again, my good students, to go through with you on a series of lectures of biology topics. And in our today's session, we will continue with the already started topic, which was growth in Form 4. And now we are going to look on the subtopics in which we will cover in our session. Considering these subtopics, if you remember, we passed through concept of growth in our previous sessions. Also, we studied about mitosis-mediated growth. Also, our last session was about growth and development stages in human. My good students, in our today's session, we are going to look on the growth in flowering plants. I'm glad you are here following me, and I know you will get deep insight on this concept of growth in flowering plants. Welcome, my Form 4 students. Starting with growth in a flowering plant, we have to know growth in a flowering plant starts from a seed to seedling that grows to a mature plant by the process of germination. And germination is the process by which a seed develops into a seedling. My dear Form 4 students, what we have to get here is that kwamba tunapozungumzia growth in flowering plants, nataka uelewe kwamba tunazungumzia flower, maua ambayo matokeo yake ni seed. Kwa hiyo kupata flowering plant tunaanzia kwenye seed. We grow or we plant seed. After planting seed, that seed develops into a real plant, and after a while that plant develops flowers. That is the real concept. Kwa hiyo kitu ambacho unapaswa kuelewa ni kwamba seed inatokana na maua. Ua linatengeneza tunda, likitengeneza tunda, ndani ya tunda tunapata seed. Hizo seed zinatumika kama mbegu baadaye kwa ajili ya kupata mimea mingine na kadhalika. So when you talk about growth in flowering plant, we are going to look on the way the seed develops into seedling through the process of germination. And the meaning of germination is that the process of seed to develop into seedling. Keep on watching, keep on following. I hope you will understand well. Let's go. The seed consists of the following parts. Before we get deep in our session, you have to know what's made up the seed. Seed consists of the following parts. The first one is an embryo. An embryo is a this part consists of radicle that develops into a root and a plumule which develops into a shoot after germination. Kwa tunavyoangalia parts za seed, my dear students, tunaenda kuangalia ile seed ambayo tutaiplant baadaye tulete flowering plant. Ina possess vitu gani? The first one tumeona ni embryo. Ambako embryo ina kazi nyingi. Hiyo ndio inayobeba maana halisi ya ule mmea kuja kukua. First inatusaidia baadaye kuja kututengenezea the roots, inatusaidia kututengenezea plumule, we shall see, as well as the shoot, as we see here. It develops into a root and a plumule which develops into a shoot after germination. Also, we have another component which is a seed coat. This part is also known as tester, which is a protective layer surrounding a seed. My dear students, until now tumeshaona kwamba tuna parts kama hizo. Hii ya pili hapa tunaita seed coat. The function of seed coat, it function as you can say a tester. Ina surround the seed for protection purpose. But embryo, as we see, kwamba its main function ni kwa ajili ya kwenda kutusaidia kudevelop into roots, and plumule ambako hii plumule ndo itadevelop itutengenezee shoot after germination. Now tunaweza kuangalia another component which is cotyledons. This part grow to form embryonic first leaves after germination. It store and provide food for young plants. Tunapozungumzia cotyledons, hope umeshasikia kuhusu cotyledons. Cotyledons, my dear students, ni kama gala. Ni kama gala. Na hii cotyledons inatusaidia kwa ajili ya kustoa chakula ambacho kitasaidia young seeds. Yaani tuseme ule mmea mdogo ile kuweza kudevelop during germination. Inastore chakula kwa ajili ya kusaidia development of young planties. Also, seeds with one cotyledon are known as monocots, as well as seeds with two cotyledons are known as the dicots. Okay. Tunapozungumzia cotyledon tumeona kwa upana kwamba cotyledon ndo kama hivyo inakuwa ni kwa ajili ya kututengenezea zile first leaves after germination. Also, cotyledon inatusaidia kutunza chakula kwa ajili ya young planties. Another thing we have to take note on this point, on this part of the seed, is that seed consists of varieties of kinds. We may have monocotyledons and dicotyledons, depending on the number of cotyledons they possess. For instance, we can see here seeds with one cotyledon are known as the monocot, and those with two cotyledons are known as dicots. A good example of monocot seeds is a maize seed. When you talk about cotyledon ni magala mahifadhi chakula. Maize, muhindi, una one cotyledon. Lakini tukizungumzia mfano wa dicotyledons zenye cotyledon mbili, we can see beans seed. Ukichukua harage, ukiligawanya mara mbili utapata pande mbili. Zile pande mbili, hiyo ni cotyledon ya kwanza, hiyo ni nyingine. Na aina nyingine mbalimbali za mimea ambazo zina fall under dicots and monocots. You should explore more so that you can deepen up your understanding. Also, we already talked about this, that seed have two cotyledons, they are known as dicots, and now we are going to look on the another part of the seed, which is micropyle, and this is a pore through which water enters the seed during germination. Also, another part of the seed is micropyle. Micropyle ni kama pore ambayo water ina pass through the seed kipindi cha germination. Na hii ni muhimu kwa sababu itasaidia ile seed kuweza kuwa soften up na kuallow permeability as well as soften the seed coat for easy germination. Also, we have hilum, another part of the seed. In this is a scar that shows the seed's point of attachment to the ovary wall. As we can see, hilum ni kama a scar ambayo inaonyesha attachment ya ovary kwenye seed. If you pass, tuchukue mfano harage lina sehemu ambayo inatoa. Yaani hilum hii tuifananishe na ile ya kwenye kidney. Hope mmesoma about gaseous exchange and respiration. Tunapozungumzia hilum pia inapatikana kwenye kidney. Tuchukue harage tuliplace kama kidney. Utaweza kupata sehemu ambako ile sehemu ambako ina hilum, alafu angalia kwenye harage, you will see the hilum, and that area is very very important for the attachment of the ovary, attachment point to the ovary wall. Also, we can see that the structure of dicot and the monocot seed, we can see in this figure below. This is the monocot. Okay, good. Here we can get all the components of our seed. For instance, here we were talking about the hilum. We can see here, this is the scar, which shows the attachment of the ovary wall. We have this micropyle, also we saw it. We have seed coat. When you open it, you can see a lot of parts, plumule, which develops into the shoot after germination. Also, we have radicle, cotyledons, and so forth. This is the dicot, also in open ways. This is the dicot, and this is the monocot seed. Okay. Hope you will understand well. Now we are going to look on the changes that occur during seed germination. During seed germination, physical and chemical changes occur in a seed. Tunapozungumzia seed germination, as I already told you, that is a process in which a seed develops into seedling. And during seed germination, there are a lot of changes which occur in the seed. And those changes comprise the physical and chemical changes. And now we are going to start with the physical changes which occur on the seed during germination. Starting with the physical changes may accompany with the first swelling of the seed after absorption of water through the micropyle. My good students, as we see, a micropyle was defined as a pore through which water enters the seed. And this caused the first physical changes in a seed. Tumeona kwamba that opening inaruhusu maji kupita katika seed. Maji yakipita kwenye seed yanasababisha swelling ya seed. Seed ina absorb a lot of water due to pressure inserted by the water, inasababisha ile seed ina swell. Also, another change, physical change, is that the water absorbed by a seed softens the seed coat and activates the enzymes in the cotyledons. My good students, the role of water hapa inaenda kusaidia kazi mbili. First, kusoften the seed coat. Kwa nini isoften the seed coat? Lengo ni kwamba itusaidie kurahisisha penetration ya ile plumule kuja katika ardhi, kuja up the ground. Kwa sababu without softening that seed coat, it will be impossible. Germination will be impossible. The role of water is to soften that seed coat ili kusaidia permeability ya maji pia na kusaidia permeability ya ile pale plumule iweze kutoka itoke kwenye out of the ground. Pia another role of water here we can see that is to activate enzymes, and we know tumesoma enzymes in many times. Enzymes kazi yake ni kwa ajili ya kuactivate ama tunasema kwamba ni kuspeed up rate ya reactions ndani ya plant body, in case ni kwenye animals pia wherever. That is the main function of the enzymes. Another physical change is that this process here is followed by the bursting of the seed coat and the emergence of radicle and plumule. My good student, hii process kulainisha seed coat inaenda kupelekea physical change nyingine ambayo seed coat itabasti. Ikibasti itasaidia emergence ya radicle na plumule kwa ajili ya growth ya mmea husika. Also, we can say the meaning of radicle. The radicle is the one that forms a primary root. The radicle continue to elongate and give rise to many side roots. And the plumule give rise to a shoot. Okay, I'm glad unaelewa. Yani I'm happy kwa sababu you understand. Tunapozungumzia radicle na plumule. Radicle we can see kwamba ni ile party ambayo ina give rise to the shoot. As we see kwamba seed ikiwa kama seed haina shoot wala haina root. Kwa hiyo kuna part itabidi develop into root na kuna part inabidi develop into shoot. Part itakao develop into root, hiyo tunaita ni radicle, lakini part itakao develop into shoot tunaita ni plumule. Those things after germination. Okay. After looking all of these physical changes, now we are going to look on chemical changes which accompany the seed changes. First, it is accompanied with the physical change. Chemical changes are accompanied with the physical changes. And now we can see, first, a seed absorbs water. This is the physical change. And this physical change leads to the chemical change, which is the food stored in the cotyledon or endosperm is hydrolyzed into a soluble form. Tunapozungumzia chemical change, my dear student, we cannot talk about chemical change alafu tukaiacha physical change. Always physical change ndio inopelekea chemical change. For instance, tumeona kwamba our seed ina absorb water. Iki absorb water inaenda kusaidia solubility ya materials inside the seed na kusaidia ama tunasema kwamba ina easier the hydrolyzation process. And that hydrolyzation of food is that we refer it as a chemical change. Okay. Also, the stored food in the form of starch is converted into sugars by the enzymes called diastase. Also, another chemical change is that kwa sababu hiyo seed inahitaji nguvu, inahitaji chakula ili iweze grow up, ile stored food ambayo tunaita starch inakuwa stored in form of starch, inakuwa converted by enzymes kwenda kwenye useful product ambayo ni sugars, kwa ajili ya kusaidia mmea uweze kufanya ukuaji. Na hii sugar, my dear student, inaenda kutumika kwenye process ya respiration, inakuwa broken down into energy, ambako ile energy inasaidia ukuaji wa mimea. Good. Also, then the growing seedling readily absorbs the dissolved stored food. We see that seed ina store chakula in form of starch. Hii starch inavunjwa vunjwa into sugars na inakuwa hydrolyzed kwa ajili ya kuweza kunaurishi mmea na kusaidia ukuaji wake. That is all about it. Chemical and physical change which occur in seeds during germination. Okay. Now we are going to look on conditions necessary for seed germination. My good student, I'm here to make you aware about this concept we are passing through. Na nazidi kukukumbusha the meaning of germination. Ni kile kitendo cha wewe mwanafunzi wangu kuchukua mbegu ya maharage ukaiotesha ardhini. Sasa ile process ya ile mbegu kudevelop kuwa seedling ama kudevelop kuwa kamea kadogo ambako baadaye katatuletea mmea mkubwa, hiyo tunaita germination. Je, ni conditions gani zinahitajika kwa ajili ya huu mmea ama seed kufanya germination? Ni condition gani zinazohitajika kuifanya seed pengine uliyootesha ardhini develop into seedling? Tunaenda kuziangalia. First, we can see water. The second is oxygen gas, temperature, as well as enzymes and hormones. And now we are going to look on the first after the another. And now we are starting with the water. When you talk about water, tunajua kwamba water ni hitaji muhimu sana kwa ajili ya uhai wa viumbe. Tunapozungumzia water on the seed, tumeshaona functions zake nyingi. First of all, hiyo seed tunahitaji iweze kugrow, iwe seedling. Na ili iweze kugrow, as we know, seed ina seed coat. This seed coat is hard. It needs to be softened so that to provide permeability for the developing seedling. Kwa umuhimu wa maji kwanza ni kwa ajili ya kusaidia kusoften up ile seed coat kwa low permeability. Nyingine ni kusaidia kuactivate enzymes ambao